Hello students, welcome to EPG Pathshala. I am Dr. Chirantan Raval from Dr. APJ Abdul Kalam Government College, Silvassa, part of UT of Dadra and Nagar Haveli. Today we are going to discuss about respiratory assemblies, which is a part of a paper Carbohydrate Metabolism. Let's see what we are going to learn in this module. Our target in this module is to study the respiratory enzymes as a free-floating or a complex assembly. We will also explore the assembly of respiratory proteins. We will try to understand the role of assembly factors in assembly of supercomplexes. Mitochondria are ubiquitous organelles in the eukaryotic cells, where its primary function is to generate ATP through oxidative phosphorylation. That's why mitochondria is known as powerhouse of the cell. Mitochondria are found in the eukaryotic organisms, but they are lacking in the prokaryotic microbe. So in the prokaryotes, the same thing is carried out within the membrane, that is plasma membrane. The mitochondrial respiratory chain is composed of 5 protein complexes. So 5 protein complexes are observed in the respiratory chain complex, that are NADH, ubiquinone, oxidoreductase, which is known as complex 1, succinate, ubiquinone, oxidoreductase, that is complex 2, ubiquinone, cytochrome C oxidoreductase, that is as complex 3, cytochrome oxidase as complex 4, and ATP synthase, that is as complex 5. Complex 1 is the entry point for electron to transfer. As you know that in the respiratory chain there is a transfer of electron from one carrier protein to another. When the electrons are transferred, they generate the proton motive force, which is required to drive the synthesis of ATP. Complex 1 is the entry point for electron transfer. It accepts the electron from NADH, which are then transferred to complex 3 via ubiquinone and then to complex 4 via cytochrome C. The proton transfer that accompanies the transfer of electron creates a gradient which provides the energy required to drive the synthesis of adenosine triphosphate. There have been always two viewpoints how these respiratory enzymes exist within the mitochondrial membrane. The point over here to discuss is how they assembled, how they exist within the membrane of the mitochondria, within the inner membrane of the mitochondria. Many people believe that the respiratory enzymes as a free floating in the mitochondrial membrane with cytochrome C and ubiquinone as connecting molecule. The other predict that respiratory enzymes exist in a solid complex composed of varying ratio of different complexes associated with each other to form supercomplexes. So there are two beliefs are there. One viewpoint that free floating, another viewpoint they exist as a supercomplex. Few complex combines to form a supercomplex, while in another case free floating assemblies of the protein is there. earlier viewpoint had more support and evidence backing it in past. Nowadays, we have advanced technologies, we have advanced biosensors. The availability of sensitive biochemical assay is slowly replacing the textbook model of independent complexes floating in the inner mitochondrial membrane in favor of mitochondrial respiratory complex model, which composed of stable supercomplexes operating as the functional unit of respiration. So as in now we are getting more and more result with the use of advanced technology, and we now slowly believes that the MRC model is having a more evidence as compared to the free floating theory. The importance of supercomplexes in the pathology of various human diseases slowly become apparent, with reduced and displaced supercomplexes being observed in various genetic, aging and neurodegenerative disorders. The concept map. In this lecture, we will first discuss briefly about the respiratory assemblies, oxidative phosphorylations and assembly factors. We will also learn about the generation of ATP through the electron transport chain. We will then discuss the assemblies of respiratory complex. Mitochondria generates ATP through oxidative phosphorylation in eukaryotic microorganisms. Oxidative phosphorylation is the process whereby the free energy that is released when electrons are transferred along the electron transport chain, which is coupled to the formation of adenosine triphosphate from adenosine diphosphate and inorganic phosphate. In intact mitochondria and in special preparation of submitochondrial particles, the transport of electrons and the phosphorylation of ADP are tightly coupled reaction. In damaged mitochondria, respiration may occur which is unaccompanied by oxidative phosphorylation. When this happens, the mitochondria are said to be uncoupled. Oxidative phosphorylation is the main source of energy in aerobic cells. In uncoupled mitochondria, because electron transport may still occur, free energy may still be released as the electrons are transferred down the transfer chain. However, this energy is not trapped as ATP and appears instead as heat. So, when it is uncoupled, we lose the energy in the form of heat instead of generation of ATP. So, I think now you get the point of coupled and uncoupled oxidative phosphorylation. If the electron transfer is not coupled with the ATP generation, then we may lose the energy in the form of heat. What is the role of mitochondria? Mitochondria plays an important role in satisfying the requirement of cellular energy through oxidative phosphorylation. Apart from this, it plays many other important roles. This important role includes buffering of the cytoplasmic calcium. It creates and liberates the reactive oxygen. Mitochondria liberates metabolites that control the crucial pathways such as succinate and alpha-ketoglutarate. Mitochondria helps in controlling the apoptosis. Mitochondria assist the cell to adapt changes in substrate availability by signaling the pathways. Mitochondria modify their organization and dynamics for quality control. Role of mitochondria indicates that it plays a vital task in maintaining the balance within the cell. It requires a strong regulation of mitochondrial activity through expression, transcription and translation. Recent study indicates that regulation of mitochondria activity is also achieved by dynamic assembly organization of the respiratory protein complex in the inner mitochondrial membrane. The mitochondrial electron transport chain, that is mitochondrial respiratory chain, consists of 5 protein complexes. Complex 1, that is NADH ubiquinone oxidoreductase. Complex 2, that is succinate ubiquinone oxidoreductase. Complex 3, that is ubiquinone cytochrome C oxidoreductase. Complex 4, cytochrome C oxidase. And complex 5, ATP synthase. Generation of ATP by electron transport. In a number of different modes of mitochondrial metabolism, including the respiration and photosynthesis, ATP is generated by transporting electron through the chain of carrier molecules with fixed orientation in a cell membrane. Although the complexity and component of electron transport chain vary, they have a certain common features. The component of chain are carrier molecules capable of undergoing reversible oxidation and reduction. Each member of a chain is capable of being reduced by reacting with the carrier molecule that precedes it, and it is oxidized by the carrier molecule that follows it. In any specific example of electron transport chain, certain members transport hydrogen atoms while others transport only electrons. The orientation of carrier in the cell membrane is such that the hydrogen carrier transport in the direction towards the outside the cell and electron carrier transports towards the inside. Thus, at each conjunction in the chain, the hydrogen carrier and electron carrier, a proton is transported out of the cell. The cell membrane is otherwise impermeable to proton. As a consequence, electron transport traps a proton of the chemical energy released by the net reaction of the chain in the form of gradient across the membrane of a proton and electric charge. Such a gradient is termed as a proton motive force. It is a form of potential energy capable of doing work. It drives certain permease system that concentrate externally supplied substrate within the cell. It provides the energy for flagellar mediated cell motility, and it drives the energy required for the synthesis of ATP from ADP. The synthesis of ATP at the expense of proton motive force is catalyzed by a complex membrane bound enzyme, ATP phosphohydrolase. Sometimes it is termed as ATPase. It is composed in all bacterial studies of two multi-component protein, that is BF0 and BF1. The subunit composition of this membrane insertion of BF0 and BF1 are referred in the figures. The alpha and beta subunits of BF1 are arranged alternately to form a hollow hexagon, the central hole of which contain the gamma subunit associated with the other subunit, that is delta and epsilon. The BF1 probably has the subunit structure of alpha 3, beta 3, gamma, delta and epsilon. The alpha and beta subunits form the catalytically active portion of structure, the site where ATP is synthesized from ADP and inorganic phosphate. The gamma, delta and epsilon subunit form a portion of translocating stuck and a gate that bring to the active site at the proper rate the portion that drives the reaction. The peptide form of a proton channel, though a membrane, they are highly hydrophobic, accounting for their intramembrane location. ATP phosphohydrolase catalyze a reversible reaction. ATP can be synthesized at expense of proton motive force, or in certain cases a proton motive force can be established at the expense of intracellular ATP. So friends, now let's look at the structure of a ATP synthesis, that is the complex 5. Whatever the precise mechanism of ATP synthesis takes place at the F1, F0 ATPase or ATP synthase. The mitochondrial F1 component appears as a spherical structure attached to the inner membrane surface by stalk, and the F0 component, which is embedded in the membrane. The F1, F0 ATPase is on the inner surface of the plasma membrane in bacteria. F0 participates in proton movement across the membrane, and this movement through a channel in F0 is believed to drive oxidative phosphorylation. F1 is a large complex in which 3 alpha subunits alternate with 3 beta subunits. The gamma subunits extends downward from the alpha 3 beta 3 complex. It composes part of the stalk and interact with the F0. The delta subunits also is located in the stalk. Much of the gamma subunit is positioned in the center of F1, which is surrounded by the alpha and beta subunits. The gamma subunits rotates rapidly in a counterclockwise direction within the alpha 3 beta 3 complex, much like a car's crankshaft, and causes a conformational change which drives the ATP synthesis within the beta sites. The major structural features of ATP synthase deduced from the X-ray crystallography and other studies. F1 is a spherical structure composed largely of alternating alpha and beta subunits. The 3 active sites are on the beta subunits. The gamma subunit extends upward through the center of the sphere and can rotate. The stalk, that is a gamma and epsilon subunits, connects the sphere to F0. The membrane embedded complex that serve as a proton channel. F0 contains 1 as a subunit, 2 B subunits, and 9 to 12 C subunits. The stator arm is composed of a subunit A, 2 B subunits, and the delta subunits. It is embedded in the membrane and attached to the F1. A ring of C subunits in F0 is connected to the stalk and may act as a rotor and move past the subunit of the stator. As the C subunit ring turns, it rotates the star. Traditional model versus supercomplex structure, that is a respirosome. There are two different views regarding the arrangement of the respiratory enzymes within the mitochondrial membrane. The respiratory enzyme complex were initially anticipated to be closely packed. Such packed structure assumes to provide more effectiveness in electron transport. However, this original model was gradually discarded and replaced by the fluid model. Initially, it was believed that the respiratory enzyme complex exists as a free-floating structure within the mitochondrial membrane. Over here, it was assumed that cytochrome C ubiquinone act as a connecting molecule. Respiratory enzymes are diffused within the inner mitochondrial membrane. In this model, the respiratory complex are observed to be free unit set in the inner mitochondrial membrane. MRC model. Mitochondrial electron transport chain, that is mitochondrial respiratory chain, consists of 5 protein complex. Due to advance in the accessibility of the sensitive biochemical assay, mitochondrial respiratory complex is slowly replacing the traditional model, that is a floating complex in the inner mitochondrial membrane. The regular and frequent observation of respiratory complex associating with each other to form a supercomplex has gradually changed the idea of fluid state model of the electron transport chain. MRC model suggests that the respiratory chain is composed of stable supercomplexes. These supercomplexes are functional structures of a respiration. MRC model suggests that respiratory enzyme forms a solid complex. This solid complex consists of a different complex which are connected with each other to form a supercomplex. Supercomplex assembly. MRC model suggests that respiratory enzymes forms a solid complex. This solid complex consists of a different complex which are connected with each other to form a supercomplexes. The most general supercomplex recognized are complex 1, 3, complex 1, 3, 4, complex 3, 4. Complex 2 is observed to be free it is observed that small portion of the complex 2 is associated with the supercomplex 1, 3 and 4. Complex 5 observed as a dimer. However, while lots of structural evidence was presented for the existence of supercomplex, but less number of evidences was available to specify their functional implications. It is observed that complex 1 assembly is dependent on the complex 3. It is also dependent on the complex 4. It is found that complex 1 was unstable in the absence of complex 3. Lack of complex 4 also affects the assembly of complex 1. The absence of complex 1 does not affect either complex 3 or complex 2. So complex 1 has dependence on their other complexes for their formation and stability. Experimental findings suggest that first individual complexes will form. This is followed by the formation of supercomplex. Mitochondrial DNA encodes product sequentially incorporated into the individual respiratory complex. There is a small gap between formation of individual complex and supercomplex assembly. In neurospora, it is observed that assembly and synthesis of a complex 1 was very closely associated with the supercomplex formation. In mouse mitochondria, observed that partial complex 1 is formed first. Size of that complex is 830 kilodalton. Then complex 3 and complex 4 associated with complex 1 subunit with the help of assembly factor. After attachment of a complex 3 and complex 4, other subunit of a complex 1, that is NDUDS4 and NDUVFV1 is attached to form a supercomplex assembly. This indicates that supercomplexes assembly precedes the assembly of individual complex formation. Stability of complex 1 may require the association of complex 4 and complex 3. So, that indicates the interdependence of the various complexes for the stability. The consistent and repeated observation of respiratory complexes associating with each other to form supercomplexes has slowly replaced the idea of fluid state model of the electron transport chain. But while plenty of structural evidence was available for the existence of supercomplexes, mainly in the form of electrophoretic pattern on the blue native polyacrylamide gel electrophoresis or a density gradient centrifugation, but there is not enough evidences was available to indicate their functional significance. If we come to know about the functional significance of these supercomplexes, then it will be very helpful in solving the many errors of the metabolism that is related to the electron transport chains. So, functional significance of the supercomplexes is yet not identified, but as and when we have a new technology that is available, I am sure that in the near future we may come out with a functional significance of the supercomplexes. Of this spectra leads to the reduced membrane potential, ATP synthesis and overall mitochondrial function. Cardiolipin attached to complex 1, 3, 4 and complex 5 in the inner mitochondrial membrane. It has a 6 binding site for complex 3 and 4. So students, let's try to conclude what we learn in this module. Mainly our discussion lies with the supercomplex and a free-floating theory. In many books you will get mainly about the free-floating theory. But if you go through the recent literature, you will find that the supercomplex theory has more evidence. So in this lecture, we learn about the respiratory assemblies, supercomplexes and their role in respiration and mitochondrial function have surfaced only recently. Yet the functional significance of this supercomplexes is yet to identify. But the role of supercomplex assembly defects in diseases such as Barr syndrome, aging and other complex 1 deficiency disorders such as cardiac myopathies, and neurodegenerative disorders is slowly coming to light. And it is of biomedical significance that the assembly and function of our supercomplexes is understood clearly. If we understood these things clearly, that is regarding the benefits of the functional significance of these supercomplexes, then we may able to solve many issues related to the diseases. So slowly coming into light, and it is of biomedical significance that the assembly and function of a supercomplexes is understood clearly. Extensive studies are required to understand how altered supercomplexes translate into mitochondrial dysfunction and how this dysfunction can influence the development and the progression of various pathogens. Development of new and advance of a new technology is the next big step in understanding how supercomplexes assembles and the function and how they can cause mitochondrial dysfunction. Thank you very much.